Hi, everyone. My name is Janet, and I'm a bioinformatics master's student at the University of British Columbia and BC Cancer Genome Sciences Center. Today, I'll be presenting efficient targeted error resolution and automated finishing of long-read genome sequence assemblies. Long-read sequencing technologies, such as from Oxford Nanopore and Pacific Biosciences, have been increasingly used in genomic research over the past several years, as these platforms have dramatically developed and improved. Long reads can span several thousands to even millions of nucleotides in length, and this provides valuable long-range genomic information for genome assembly. This information aids in resolving repetitive regions, identifying structural variants, and reaching chromosome-level contiguity. However, the low accuracy of long reads remains a major limitation to their application, as this affects the accuracy of data obtained from them, such as genome assemblies. This is an issue because accurate genome assemblies are crucial for annotating functional elements and identifying clinically relevant variants. Often, genome polishing is done after long-read assembly to correct base errors using more accurate short reads. One such tool to do so is RACON. However, RACON and other similar tools mostly rely on read alignments, and this makes them memory-intensive and not scalable to large genome sizes. Today, I will be presenting an alignment-free genome finishing protocol that uses short reads to correct assemblies generated from long reads. This is a camera-based method that employs memory-efficient Bloom filters and is composed of two previously published tools, ntEdit and Sealer. Bloom filters are a probabilistic data structure used to store and test for the membership of an element in a set. In bioinformatics, they're often used to store k-mers, which are substrings of length k obtained from nucleotide sequences such as sequencing reads or genome assemblies. Bloom filters can also be used to implicitly represent de Bruijn graphs. In this representation, the nodes of the graph are k-mers that are inserted into the Bloom filter, and edges are implicitly identified by querying the Bloom filter for all possible extensions of a given k-mer. The first step of the protocol is using ntEdit to polish the long-read assembly using k-mers generated from short reads. ntEdit iterates from long to short k's to verify each k-mer in the assembly and attempts to fix errors when an assembly k-mer is absent in the Bloom filter. In brief, all mismatches in indels up to 5 base pairs in length are tested for presence in the Bloom filter, and an edit is made if there is a well-supported match. If none of the possible edits have enough support, this problematic region is flagged by soft masking for further targeting. The polished assembly with flagged problematic regions is then input into the next tool, Sealer. Sealer uses implicit Bloom filter de Bruijn graphs from short read k-mers to fill gaps and flagged regions in the assembly, also iterating from long to short k-mer lengths. Gap linking sequences are connected by performing a depth-limited bidirectional breadth-first search within the Bloom filter de Bruijn graph for a path that connects the flanking sequences. And the final output after this step is a base-corrected, gap-filled long-read assembly. So today I'll be presenting results obtained from this protocol on Shasta and red bean assemblies for two individuals, NA12878 and NA19240. The 4 baselines were all assembled with ONT Promethean reads and corrected with corresponding Illumina HitSeq reads. The protocol was run using k-mer lengths 80, 65, and 50, and the results were compared to RACON using Minimap2 alignments. Both tools were run with a total of 48 threads, and the assembly quality was assessed using Busco and Quast. ntEdit and Sealer brought all assemblies to near Busco completeness, which can be seen on the y-axis. This is an increase of approximately 24 to 50%. The protocol also reduced the number of mismatched bases on the x-axis by up to 58%. So overall, the resulting assemblies from ntEdit and Sealer are highly comparable to those produced by RACON. ntEdit and Sealer required significantly less RAM than RACON, about 1/10, using 100 gigabytes versus approximately 1 terabyte respectively. The protocol also completed in less time than RACON in 3 out of the 4 runs. Here is a step-by-step breakdown of the runtime for each tool. Sealer is the most time-consuming step for the presented protocol, but all steps take significantly less time to complete than RACON. ntHits and Abyss Bloom are the Bloom filter creation steps for ntEdit and Sealer respectively, and this is a redundant step that will be consolidated in the future, further speeding up the protocol. In conclusion, the ntEdit and Sealer protocol produces highly complete and accurate assemblies while requiring significantly less memory than RACON. This makes it a scalable and accessible solution for the targeted resolution of errors in long-read assemblies. I would like to thank all of my fellow lab mates at the BTL, but especially Renee, Lauren, Jonathan, and Ananj, who have worked with me on this project. I'd also like to thank our funding partners, Genome Canada, Genome BC, and the National Institutes of Health. Thank you so much for your attention. You can find ntEdit and Sealer at the sources shown on the slide. If you have any questions, I'd be happy to answer them in the chat, and you can also email them to me at the email listed here. Have a great rest of your day.